process of differentiation. Yet all these cells are genetically identical, simply because they're all derived from one original cell. Each mitotic division ensures that daughter cells receive an exact duplicate of all the genetic material. So each type of cell must have a different subset of the whole genome operating. Cells in this bone, for instance, have their own particular set of genes in operation. Genes are never lost, but they are switched on and off as required. These are a bacteria called E. coli. They can be grown on a variety of food sources. Each food source induces specific enzyme synthesis. But how? In 1965, two French scientists, Jacob and Monod, received their Nobel Prize. Their work in the early 50s unraveled some of the secrets of this regulation. To produce enzymes, three genes were involved, the structural genes. Close to these genes was another region, the P gene, where the RNA polymerase binds. But with this system, the bacteria would always produce enzyme, the polymerase moving down the structural genes, producing RNA templates. But Jacob and Monod had proposed another region, which according to them occurred between the P gene and the structural gene. This operator gene was the key to genetic regulation. They suggested from genetical observations that this was a site at which a repressor molecule would bind, and so obstruct the movement of RNA polymerase molecules. The repressor molecule was produced by another gene close by, the I gene. In the presence of lactose, the food source, the lactose molecule binds to the repressor molecule, freeing the RNA polymerase, and protein synthesis occurs. In the absence of lactose, the repressor is free to bind to the OG. Evidence for the lac operon was based on genetical experiments. Direct evidence could only come from the isolation of the repressor molecule, and for many years this proved to be impossible. Doubt of the validity of the model grew. Harvard University, where in 1966 a group of scientists isolated the repressor molecule. The group was led by Professor Walter Gilbert.
In a sense, our answer to how the repressor works now is very simple. At the end of all this, we know that the repressor controls the gene by binding to the DNA at a very special site right at the beginning of the gene. When that protein sits on the DNA, the RNA polymerase, another large protein, can't get up to the DNA to bind to it. However, when the sugar comes and binds to the repressor changes its structure and falls off the DNA. That leaves the DNA free. The repressor bound to the sugar has fallen off, and now the polymerase can come and stick to the DNA. It does so by binding over almost 40 bases, 4 turns of the DNA. It recognizes the DNA at this position, and over here, it separates the strands of the DNA and then begins to copy, moving along the DNA to make a copy of the DNA of that gene. What these experiments showed is that we could completely understand a controlling element in bacteria. The important part of that controlling element is that we have a gene that makes a protein that both can go to the DNA and control the second gene and bind to some other effector molecule. The protein serves as a way of translating between the effector molecule and the control of the gene. We don't yet know in any higher organism, in any eukaryotic cell, the precise nature of any of the controls. But we do believe that those controls will be exercised through proteins that go to the DNA and bind at unique sites there.
Some of our knowledge of genetic regulation of eukaryotic organisms has to come directly from the study of gene products, the messenger RNAs and the proteins themselves. The lens from a human eye, it's made up of different types of crystalline proteins. In order to maintain harmony with the curved retina, the lens has a higher refractive index in the center than at the periphery, and this is brought about by different crystallines being laid down at different times during development. The lens is built like an onion.
new fibers being produced from the epithelium as the eye grows. The first fibers contain only delta and crystalline proteins, while fibers formed later have virtually no delta crystalline. Beta protein takes its place. But what triggers proteins being switched on and off? There appear to be several. One is nearness to the retina. This causes the elongation of the cells into fibers. As cells elongate into fibers, crystallines begin to make their appearance. But this by itself wouldn't explain the gradual change with increasing age of the mix of crystallines in a cell. Lens epithelial cells, called lentoids, can be grown in cell culture. The embryo cells divide very fast. It takes much longer for the cells of an older animal to divide. It seems that the shorter cell division time is conducive to a higher proportion of delta crystalline, and the longer cell division time leads to an increase in the amount of beta crystalline. So, the control of the synthesis of different crystallines seems to be determined by several triggers: positional effects and timing. Protein levels at different stages of lens development can be monitored using the technique of electrophoresis. But do these changes in protein reflect changes in gene activity? To investigate this, we really need to look at messenger RNA itself.
Isolated messenger RNA can be used to make a complementary copy of DNA, cDNA. In the presence of the enzyme reverse transcriptase, DNA bases are joined along the messenger RNA molecule. The messenger RNA can now be removed by hydrolysis, leaving the cDNA, which has a sequence complementary to the mRNA corresponding to a specific protein. This can now be used as a probe to find this protein in tissues. It works like this. If the cDNA is mixed with messenger RNA, in time, all the cDNA hybridizes with the messenger RNA to which it's complementary. Expressing this graphically, the amount of hybridization increases with time until all the cDNA has become bound to messenger RNA. Using non-complementary RNA, however, no binding takes place. If now a mixture of RNA is used, as in the normal experiment, it'll take longer for the cDNA to hybridize because there's less complementary mRNA in the mixture. If more complementary mRNA had been present, the hybridization time would have been faster. So this method provides a way of looking at messenger RNA production in different lens cells.
Some animals, like newts, have the ability to regenerate a missing lens from the iris, a totally different tissue. So some cells have a capacity to develop into several products in the embryo. When they do, is the change reflected in the mRNA turnover? By using cDNA to lens crystalline mRNA, it was possible to show that the cells of the very early retina of the chick embryo actually contain crystalline message, although they're going to differentiate into retina and not into lens. As the retina differentiates, the capacity of these cells to turn into lens in culture gradually diminishes. In parallel with this, the amount of chick lens crystalline mRNA declines. The number of cells that can turn into lens diminishes, and the amount of messenger present drops, until finally their ability to turn into lens is totally lost. The way in which genes are switched on and off is one of the central questions in developmental biology. The way in which DNA is packaged into chromosomes may be of critical importance here. One of the secrets of packing centers around a structure known as the nucleosome. The three-dimensional structure of a nucleosome has been determined by X-ray diffraction. This is a simplified model. Dr. Aaron Klug, MRC, Cambridge.
First of all, the DNA is a long molecule, very long. If all the DNA of a chromosome were stretched out, it would reach a few centimeters long and has to fit into a space of only a few microns. Secondly, the DNA is a rather stiff molecule, but it can be bent. And indeed, this is the clue to the first stage of folding of DNA in a chromosome. In a chromosome, the DNA is combined with a set of basic proteins called histones. And 4 of these come together in pairs to form a rather compact particle which has the shape of a helical ramp. And on this helical ramp, the DNA is wound. It's wound into 2 turns. This contains about 200 base pairs of DNA, so that the condensation we've arrived at is something like a factor of 7. Now, there is a 5th histone called H1, which fits on to the ends of the stretch of DNA and clamps them tightly to this particle, which we call a nucleosome. It seals it off, as it were, so it is comparatively stable. Now, at the same time as this stretch of DNA is being condensed by this nucleosome histone aggregate, there is successive stretches also being condensed. And these link together through the association of their H1s, and they are connected in a continuous chain through a linker of DNA, which is short, to form the basic chromatin fiber. This has a diameter of about 100 angstroms and has a condensation ratio of 7. Well, this fiber is flexibly jointed because the histones have converted the apparently intractable problem of packaging a long, stiff molecule into that of packaging a set of separate beads, bead-like entities. And this is the entry to the next level of folding, for which we have to change the scale of our models. In this model, each nucleosome is represented by one of these flat discs with the 2 turns of DNA represented on it. And in the second level of folding, the 100 angstrom fiber is wound into a superhelix in which the turns come close together so that we call it a solenoid, forming a fiber of about 300 angstroms diameter. In this solenoid, there are about 7 nucleosomes per turn of the helix, so that we now have a condensation ratio of 7 times 7, giving a total compaction of about 50. Now, while this might represent roughly the state of condensation in a interphase chromosome, we've sort of a good deal off from the much more condensed state of a metaphase chromosome. Now, we don't know how this is achieved in detail. We can have a reasonable guess at how this is achieved. And again, the clue to the further condensation comes from the flexible nature of the chromatin fiber. The solenoid, which is made of a flexible chain of beads, can easily be bent to form a loop. And in this loop, we now have formed a fiber of about 500 angstroms diameter.
These theories are supported by an elegant but simple experiment. A metaphase chromosome can be progressively relaxed by removing the histones entirely. Only a few non-histone proteins remain attached to the DNA. examination of the structure in the electron microscope shows a remarkable feature. The overall chromosome structure remains intact. The chromosome is simply expanded, apparently held together by a central structure. The DNA has spilled out, but in micrographs it's clear that the chromosome structure has not been completely lost. Loops of DNA can be traced back to the central scaffold, and we find that the loops start at one point in the scaffold and return to a point near its origin. Could it be that these loops, when compacted by the histones, form the 500 angstrom loops we postulated earlier? Supporting evidence comes from thin sections of intact chromosomes, where the histones haven't been removed. The fiber in these chromosomes appears to be radially distributed, attached to a central axis. Projections are formed by loops made up of the basic nuclear protein. The fiber has the diameter of 250 to 300 angstroms, and the loops have the expected diameter of 500 to 600 angstroms. The loops can be seen when we examine the surface of chromosomes. The projections are compacted loops. Professor Lemley, Princeton University.
The experiments discussed suggest that chromosomes have a central axis. This axis goes from the top of the chromatid all the way through to the other end. The DNA in this case is attached, forming loops. We've seen these loops in the histone-depleted structures. They're arranged in a radial way, probably hooked together and linked together by this set of non-histone proteins. The job of the histones, then, is to compact this loop by a factor of 40 down to shorter loops. These loops get fatter now because they're covered with histones, and they have about a length about 0.6 microns, where the extended loop has a length of about 23 microns in length. So now we like to think that these compacted loops are arranged in a radial fashion, forming a kind of a rosette. This rosette goes progressively around in a helical fashion around the center of this axis, up from one end of the chromosomes all the way to the other end. So by this simple model, we suggest it's possible to explain the high compaction ratio observed in metaphase chromosomes very neatly. So it appears that the chromosome structure may not be as complicated as we thought initially. The job of the histones is to compact the DNA by factor of 40, while the job of these non-histone proteins is to tie these fiber into loops and arrange them in a radial fashion around the central axis of the chromatid.
Putting all this information together, it appears that the naked DNA is first condensed around the nucleosome core histones. The string of nucleosome units then coil to form a solenoid structure, which is bent into compact loops. Finally, these are attached to non-histone proteins that form the central scaffold of the chromosome. The tightly packed chromosome goes some way towards explaining why only certain sections of DNA are transcribed in any particular cell. In most cells, over 90% of the DNA remains tightly folded throughout the cell's life. Before a section of DNA can be transcribed to form messenger RNA, the compact loop has to be unfolded to form the bead-like 30-nanometer fiber. RNA synthesis can now begin with the help of enzyme RNA polymerase. But in this bead-like fiber, the DNA is still tightly wrapped around the nucleosome. So how can RNA polymerase get in there to transcribe it?
One of the keys behind this must lie in the internal structure of a nucleosome, or in other words, in the essential properties of these histones making up the nucleosome core. When we come to consider active genes, we are of course thinking of the chromosome not in its condensed metaphase state, but in its interphase state, where the chromatin fiber is decondensed, and depending upon the type of cell, many of the genes will still be inactive, and we think that they are probably locked up still in the form of a 300 angstrom fiber. But the active genes must be much more open than that. And here, there's no doubt that we have something like a chain of open nucleosomes from which the H1 is missing. But the great surprise has been that the other histones are still there. They don't go away. Well, then how does the polymerase manage to read through? Well, we don't know the answers to this, but there are experiments in the test tube. And here we must change the scale, go back to the original scale of our nucleosome. Here you see the two turns of DNA. And the experiments show that under certain rather mild conditions, the nucleosome can be opened up into some kind of linear structure in which the histones are probably still hanging on by their tails, but the DNA is much more accessible, so the polymerase can read through and so perform its function of active transcription.